Today we're going to learn about mitosis
and meiosis.
Mitosis assists in growth, repair, and maintenance of somatic body cells. It passes on genetic information that is identical to the parent cell. The cell's genome is passed on in the form of chromosomes. The cell carries out this process with the cell cycle. The mitotic cell cycle alters between the mitotic M phase and interphase. Interphase is divided into three parts: G1, S, and G2. G1 is the first gap because it represents the first growth phase during which the cell has made the decision whether or not to divide. S phase or synthesis phase is when chromosomes are replicated to form sister chromatids. Sister chromatids are two copies of a duplicated chromosome that are attached at the centromere. Two sister chromatids make up one chromosome. After this occurs, the cell continues to grow during G2 phase or the second gap. After G2 is completed, the cell is ready to divide. Prophase is the first part of the M phase. The DNA begins to change from the form of chromatin into a more condensed shape within the nucleus, where the sister chromatids can begin to be seen. Here, the mitotic spindle, which is made up of microtubules, begins to form, which is responsible for pulling apart of chromosomes and the cytoplasm of the cell. The two centrosomes begin to migrate to different poles within the cell. Here, the nuclear envelope, which holds the DNA, disintegrates, allowing the microtubules to extend from each centrosome and disperse throughout the cell. The microtubules attach to the proteins around the centromere of the condensed chromosomes called the kinetochore. All other microtubules that have not connected to a kinetochore attach to one another to aid in the pulling apart of the entire cell. In metaphase, the chromosomes line up with their centromeres on the metaphase plate. The metaphase plate is an imaginary plane in the middle of the cell. Each chromosome has two microtubules, one from each pole, connected to the kinetochore so that each sister chromatid is pulled to the opposite side. Anaphase is when the microtubules utilize ATP to break the cohesions that held the sister chromatids together to pull apart the chromosomes. The cell elongates as the microtubules shorten and the chromatids migrate towards the poles. In telophase, two nuclear envelopes begin to form on either pole of the cell to enclose the genetic information. Chromosomes become less condensed as they begin to form back into chromatin. Then the microtubules disintegrate and are recycled for the next loop of the cell cycle. Telophase marks the completion of mitosis. After comes cytokinesis. In animal cells, the cytoplasm is cleaved to form a furrow, pinching the cell into two. In plant cells, a cell plate forms to divide the two daughter cells. At the end of mitosis, two daughter cells that are genetically identical to the parent cell have formed from one division. The number of chromosomes in each daughter cell are the same as the parent, also known as diploid. In humans, there are 46 chromosomes in the parent cell and the two daughter cells.
The ability of cells to undergo meiosis allows for organisms to reproduce members of their own species. In order for the human life cycle to take place, fertilization occurs between the two gametes, reproductive cells from the mother and the father. Both the sperm and the egg are haploid, meaning there are half the number of chromosomes in the gametes than in the somatic cells. In humans, the haploid number is 23. Interphase in meiosis is identical to that of mitosis. In this phase, the chromosomes begin to condense, the centrosomes move towards the poles, spindles form, and the nuclear envelope breaks down as in mitosis. However, paired homologous chromosomes pair along their lengths instead of sister chromatids. Homologs are a pair of chromosomes that possess genes for the same characteristics at corresponding loci, entering a state called synapsis. Synapsis allows for crossing over to occur. Crossing over is the exchange of corresponding DNA molecules between non-sister chromatids and is one of the causes of genetic diversity. The places in which crossing over has occurred is called the chiasmata. Microtubules attach to two kinetochores, one for each homolog. The pairs of homologous chromosomes line up on the metaphase plate. The microtubules shorten to pull apart homologs towards their respective poles, elongating the cell. Sister chromatids remain intact at the centromere. The chromosomes during telophase 1 look similar to the chromosomes at the beginning of mitosis. However, the chromatids are non-sister and are not genetically identical, and each cell has a complete haploid set of chromosomes. The remainder of the cell cycle looks very similar to mitosis, as meiosis 2 separates sister chromatids. There is no replication between meiosis 1 and meiosis 2. Meiosis II takes place in both of the two daughter cells that were created by meiosis I. The positioning of chromosomes on the metaphase plate allows for another form of genetic diversity, as the chromatids are not genetically identical, and therefore the daughter cells will be genetically different from one another. Anaphase II, telophase II, and cytokinesis look identical to that of mitosis. At the end of the two divisions, meiosis I and meiosis II, 4 daughter cells will have formed. All 4 of the daughter cells are haploid, meaning they have half the number of chromosomes as the parent cell. The daughter cells are genetically distinct from the parent cell and from each other.